Hi everybody, welcome to part 4 of the cell structure and function module. We're going to talk about the organelles that make proteins. So this is going to include ribosomes, which are the protein making factories, the endoplasmic reticulum, Golgi apparatus, and transport vesicles, which are part of the endomembrane system, and we'll finish up talking about lysosomes. So once again, here's an overview of a generic animal cell. We're talking about these organelles that are between the nucleus, so the endoplasmic reticulum, the Golgi apparatus, transport vesicles. Here's a little lysosome down in the bottom. So these are molecules and structures that are involved in making proteins. So let's start with talking about ribosomes. Ribosomes are the little factories that make proteins. They take information from the DNA that's converted into a messenger RNA molecule, mRNA, and the mRNA binds to the ribosome and is used to make protein. Ribosomes have a large subunit and a small subunit. Each subunit can come apart. Any large subunit can interact with any small subunit. They're interchangeable parts. Each subunit is also made up of a variety of different proteins and different RNA molecules, ribosomal RNA or rRNA. There appears to be two different pools of ribosomes in the cell, but really they're the same ribosomal subunits. It's just depending on what type of protein they're making. So a ribosome can be free or it can be bound. All right, so free ribosomes are these dots that are floating in the cytoplasm over here, and they're bound to mRNAs that are making hydrophilic proteins. Other ribosomes are bound to the endoplasmic reticulum membrane, specifically the rough endoplasmic reticulum. We'll talk about that in a minute. And they are making proteins that are going to be either membrane proteins, so they need to be inserted into a membrane as they're being synthesized, or they are going to be secreted outside of the cell, and so they need to be inserted into the interior of the endoplasmic reticulum. Okay, so let's talk about the endoplasmic reticulum. There are two parts of the endoplasmic reticulum, but they're really connected with each other. And that's the rough endoplasmic reticulum, or RER, and the smooth endoplasmic reticulum, or SER. Now remember that the endoplasmic reticulum membrane is continuous with the nuclear envelope. So here we've got the nuclear envelope with all the chromatin and DNA removed, just to show that there are places where the outer part of the nuclear envelope is connected to the endoplasmic reticulum. There's a connection there. There's a connection there. Hard to see if there's any other connections in this picture. But anyway, but although they are connected, there is still a barrier, the inner membrane of the nuclear envelope. So material cannot move from the nucleus into the lumen or the interiors of the endoplasmic reticulum separated from the cytoplasm. They cannot move between those. The rough endoplasmic reticulum is shown over here, and you can see these little red dots. Those are ribosomes, and so the rough ER is rough because when you look under the microscope, you see these little bumps due to the ribosomes, so it looks rough. And smooth endoplasmic reticulum, it's connected to the rough endoplasmic reticulum, but there's no ribosomes, so the membrane looks smooth. The rough endoplasmic reticulum is specialized for synthesizing secreted proteins and membrane proteins, and the smooth endoplasmic reticulum is specialized for lipid synthesis and membrane synthesis. Okay, so but both parts are involved in synthesizing molecules, macromolecules. I want to point out that material that is made by bound ribosomes is enclosed into transport vesicles. We're seeing a transport vesicle over here for transport to the next part of this endomembrane system. excuse me, the next part of the endomembrane system is the Golgi apparatus. So here is a transport vesicle coming from the endoplasmic reticulum. There are some molecules inside here. These would be proteins that are soluble. Remember, there's also going to be proteins that are embedded in the membrane. Those would be membrane proteins. But this transport vesicle fuses with the membrane of the Golgi apparatus so that its contents can become part of the interior portion of the Golgi apparatus. Now, each of these stacks of membranes in the Golgi apparatus is going to have different enzymes in it that are going to modify these proteins that are being synthesized. So they might add sugars, they might modify sugars, they might add different types of functional groups to the proteins, but the processing is what is happening in the Golgi apparatus, and it appears that material moves from stack to stack of the Golgi apparatus through transport vesicles until it gets to the end, and the material that buds off into these transport vesicles can then go to their final destination, which might be secreted outside the cell. It might be being part of the plasma membrane. It might be part of the membrane of another organelle. It could be a lysosome. It could be any number of different places. So I just wanted to sort of summarize and remind you where proteins are made. Remember that the free ribosomes are making proteins that are going to be in the cytoplasm, and they are going to be hydrophilic, water-loving. But the proteins that are made on the ribosomes that are bound to the endoplasmic reticulum are going to be either membrane proteins, proteins that are going to live in some membrane inside the cell, or they're going to be proteins that are secreted outside of the cell. So they're made on the ribosomes bound to the RER, or the rough endoplasmic reticulum. They get modified and sorted in the Golgi apparatus, and eventually they are either secreted or inserted into a membrane of an organelle somewhere in the cell. And there's lots of places they can go. I just want to talk about one potential destination for proteins, and that is lysosomes. And I wanted to talk about lysosomes rather than any of the many other organelles that we could talk about, because lysosomes are very important in your cells. They have an acidic content, very low pH, and the enzymes that live inside lysosomes work best at low pH. Now remember that the pH in most of the cytoplasm of your cell is very close to neutral, close to 7. And so these acidic spaces inside your cells are really important because these are the organelles that digest material. All right, in the process of phagocytosis, there are something that's coming from outside the cell, and here it's a little piece of food that's engulfed. This food vacuole fuses with the lysosome, and then the combined structure has a lower pH than the cytoplasm. These enzymes digest this piece of food into small parts that can be used by the cell. So proteins are broken down into amino acids, and sugars are broken down into monosaccharides, right? The complex carbohydrates. Fats are broken down. Whatever the molecule is, it's broken down so the cell can reuse those things. Lysosomes can also break down contents from inside the cell in a process called autophagy, self-phagy eating. So for instance, other organelles, here is a peroxisome and a mitochondrion. They're just a couple of organelles that were in the cell. They have worn out. They've been used for a long time. The cell is not getting use out of them anymore. So they're enclosed in a vesicle which fuses with the lysosome. And again, even these organelles, these large structures, can be broken down into their individual components, and the components recycled and reused. All right. So it's really important that you have this process and this ability to break down these molecules. You don't want these enzymes just loose running around in the cytoplasm because they might start digesting the wrong structures. So by making them only function at low pH so that they have to function inside these vesicles, these structures, your cell can regulate and only digest structures that have been worn out. And in fact, there are a lot of diseases, I'll just point out, that are due to problems with lysosomal enzymes. And so it's due to mutations because the enzymes that digest the sugars or the lipids just aren't working right. So this is an important process. Okay, so this was a description of how proteins are made. Here are your terms and concepts to know so that you can create your study guide, and I will see you in the next video in this cells structure and function module. Bye.